All right, so hello everyone, and welcome to the first digital neuropathology webinar session of the series. I'm Dr. Maggie Flanagan, a neuropathologist, assistant professor of pathology and neuropathology core leader at Northwestern University, and I'm very excited to introduce our speaker, Dr. Charles L. White III from UT Southwestern today. He is a professor of pathology, director of neuropathology, and the director of the Winspear Family Special Center for Research on the Neuropathology of Alzheimer's Disease. Today, he will be presenting on the history and overview of digital pathology, which will be followed by a session with our panelists. So following Dr. White's talk, I will just be giving some brief introductions of the panelist members, and then we will have a short question and answer session at the end. So take it away, Dr. White.
Thank you very much, Maggie. I'm honored to be able to get this session started with this, what will be, I think, a very brief overview of the history and development of digital pathology with specific reference to neuropathology and, more importantly, to neurodegenerative disease. I have no disclosures. So I think we would all agree that we live in a digital world. Microprocessors dominate everything in our lives, from our thermostats on up to the work that we do every day in our careers. This digital revolution, as I like to refer to it, has occurred over about the past 40 years. And I wanted to go through some of the background changes that came into our lives that characterize the creep of digital media and digital technology into virtually everything we do. So I was a teenager in the 60s and 70s, and when I grew up listening to music, we had phonographs and we had vinyl LPs. And that was really that was really it until a little bit later on, when another analog medium, magnetic tape, became available in the consumer market and really added an element of portability to music that wasn't available with LPs and turntables. The first digital recordings came into the consumer market in 1982. And this was the compact disc or CD format. And then later on into the early and mid 90s, we saw the development of MP3 players. And probably the most popular among those was the Apple iPod. And in fact, the delivery of music in the MP3 format and the production of the iPod is said to be what saved Apple Computer from bankruptcy. And of course, nowadays, digital media and digital music are really the standard, and digital streaming of music and other content to our homes is something that we all rely on. Now, besides the music industry, another field where digital technology really took hold, and one that's critically important to the facilitation of digital pathology, was the development of photographic image recording in the digital format. In the era of film, a roll of film, 36 exposures generally, was cost in the range of $15. This was the film cost itself, as well as the price of development. Currently, you can buy many sizes of SD cards, but a 32 gig card these days retails for about $9, which can store 3,600 photographs. And of course, these are reusable. Another critically important development in the digital realm that has facilitated the development of digital pathology is the cheapening of storage in the fixed format or hard drive format. In 1983, you could buy an IBM PC-1, the first of the desktop computers that was really popular in the United States. And you could get a 10 megabyte hard drive for an extra $1,000. Today, you can buy a 1 terabyte hard drive for roughly $45. I like to do the math on these kinds of things. If you were to buy that $1,000 hard drive that cost $1,000 in 1983, today it would be a fraction of a penny for that same amount of storage. Whereas that 1 terabyte hard drive that we can currently purchase for $45 in 1983 costs would have been $100 million. Needless to say, way out of the range of anybody's ability to purchase. So clearly one of the areas that preceded the development of digital pathology was the evolution of digital radiology. Digital radiology first made its way into the scene in the form of computerized axial tomography or CAT scanning, nowadays more commonly just called CT or computerized tomography. The CT scanner, first CT scanners were developed by a fellow named Godfrey Hounsfield. Hounsfield was working at a company in London called Electrical and Music Industries, or EMI. And Hounsfield himself shared the Nobel Prize with Alan McCormick, who was more of a mathematician in 1979 for the development of this technology. So Hounsfield was an engineer with EMI. EMI, coincidentally, is also the same company that recorded all of the Beatles music and many other important bands of this era. There's some apocryphal stories about Beatles record revenues essentially underwriting the cost of the development of CT, but as much as a consummate Beatles fan as I am, that I'd like to believe that the majority of the money that went to EMI to develop the CT scanners was from the British equivalent of our NIH. So that was really public funds. And I should point out that you may in the older literature see the term EMI scan or EMI scan. And again, since the first CT scanners were produced by EMI, the first CT scans were often called EMI scans. And on the left here, back in the 1970s, it wasn't uncommon to see really poor quality by today's standards, Polaroid prints of CT images in patients' charts. On the right, we see what later evolved in the CT realm, and that was the actual printing of digital images onto radiographic film. And that was done for several reasons. Number one, radiologists didn't all at this point have high quality desktop computers where they could visualize these images. And secondly, radiology departments were still full of film readers and they knew how to look at images on film. So these digital images were transferred to really an analog medium, which is radiographic film. That eventually gave way to the availability of large, high quality medical grade monitors throughout radiology departments and development of image storing capacity in the form of picture acquisition and communication systems or PACS systems, the development of radiology software like Images on Call, so that radiologists could read films anywhere that they were sitting that had access to a computer. And of course, every day, neuroradiology these days has the capacity to do voxel-based morphometry and a number of other analytical techniques on these digital images. So radiology has pretty much made the full transition to a digital medium. So enter digital pathology. When we think of the way that we diagnose diseases, probably the first and foremost most common mechanism used by pathologists is that of pattern recognition. We recognize discrete processes based on templates that we've learned during our training and experience. Examples of this are abscesses and metastatic carcinoma. Even the most minimally trained pathologist is able to look at a histologic slide and quickly recognize an abscess or a metastatic carcinoma because of their characteristic combination of features. But in neuropathology in particular, we deal with a lot of other kinds of processes that I'll refer to as continuous processes. One of the prime examples might be glioma progression, where we go from normal to maybe a reactive appearing brain histologic appearance on up through various grades of histologic atypia in the development of eventually glioblastomas. Another common area and important for this talk today is the characteristics of neurodegenerative disease. Most commonly, we would think in this realm about Alzheimer's disease, where we know that there's a progression from the features that we see in so-called normal aging on through fully developed Alzheimer's disease neuropathologic change. And so one of the questions that arose early on in the recognition of these continuous processes was, how can we characterize these more objectively, and more importantly, how can we quantify these changes? So there were some papers that came out many decades ago that are still landmark papers in today's realm. Two of these were back-to-back papers in 1970 and 1968 that were published by a group from Newcastle, consisting of Bernard Tomlinson, a neuropathologist, Gary Blessed, and Martin Roth, who were clinicians. And they had the hypothesis that the degree of neuropathologic change of the type that we now recognize as Alzheimer's disease neuropathologic change would correlate with the degree of clinical disability in patients with cognitive impairment. And so they wrote very nice descriptive papers that should be read, I think, by any neuropathology trainee these days, looking at the correlation of neuropathologic features with clinical cognitive impairment. And in 1997, the International Journal of Geriatric Psychiatry reviewed these papers in a series of key papers in geriatric psychiatry that they wanted to bring attention to. And in the article that describes these two papers in the 1997 review is an excellent interview with Bernard Tomlinson, asking him about the history of how these ideas that they had developed and how they decided to measure what they measured. Because what they ended up measuring was the density of neuritic plaques. And I love the quotes by Bernard Tomlinson in this paper. With reluctance, we decided not to attempt actual counting of neurofibrillary tangles, that is, but to describe tangles merely as present or absent, or present in small, moderate, or large numbers, patchy or widespread in the cortex. And the other comment that he made that I think was really humorous, and if I could do a British accent, I would read it in that. But he said, counting was never a procedure calculated to lift the spirits, particularly as it had to be done after the normal day's work. And remember, they didn't have computer-assisted morphometry. This was all manual counting techniques. And one of their key papers, published in 1968, was called The Association Between Quantitative Measures of Dementia and of senile change in the cerebral gray matter of elderly subjects. And again, this paper looked at the density only of neuritic plaques in the cerebral cortex and correlated that with cognitive impairment. Well, the importance of this observation can be underscored by the fact that from this point up till 1997, when we had the NIA Reagan criteria, the neuropathologic diagnosis of Alzheimer's disease was strictly a plaque-based effort. And really neurofibrillary tangle density didn't come onto the scene until after the Brox published their important papers on the pathologic criteria, on the pathologic features of aging, and on the adoption of the NIA Reagan criteria that took into account not only plaque density, but also Brox stage as a major measure of neurofibrillary tangle density and distribution. There were early papers that looked at various quantitative aspects of Alzheimer type neuropathology, and a key paper published by Bill Marksberry at the University of Kentucky using computer-assisted image analysis brought attention to the fact that neuropil threads in the cerebral cortex in people with Alzheimer's disease were really an excellent correlate of the degree of cognitive impairment, as were neurofibrillary tangles. And in fact, they pointed out that neuritic plaque density wasn't really a very good correlate after all of the degree of cognitive impairment. And I think we still regard today the density of neurofibrillary tangles and of dystrophic neurites, and not so much amyloid burden or senile plaque density as the most important neuropathologic correlates of cognitive impairment. Another important computer-generated set of data came from the Terry, Maslia, Katzman group in UC San Diego, where they used immunostain sections stained with an antibody to synaptophysin and computer-assisted video densitometry to look at the density of synapses in the neocortex in subjects with Alzheimer's disease. And they also found that synapse density was correlated with the degree of cognitive impairment in Alzheimer's disease. And they actually suggested that synapse loss was the basis for cognitive impairment in AD, a hypothesis that is still very actively viable today. Now, in the early 2000s, robotic microscopes and whole slide imagers began to make their way onto the scene. One of the earliest commercially available whole slide imagers was the Nikon CoolScope, pictured on the left here, which would take a single slide and digitize it and produce an image that you could look at on a computer screen. About 10 years later, other whole slide imagers, such as the Aperio imager seen on the right, began to make their way onto the scene. And I would say that in today's world, whole slide imagers are really considered to be synonymous with digital pathology. So anytime you ask anybody what they mean by digital pathology, it will almost always conjure up an image of whole slide imagers. And just to show how well this technology has developed in a relatively short period of time, on the left is a scan made on a Nikon CoolScope. This took 2 hours at 5x. An Aperio scan, on the other hand, pictured on the right of that exact same slide in 2013, took only 3 minutes at 20x. And at 20x, you get a whole lot more information than you get at 5x that can be used then for further analysis. So one could ask, why did pathology lag a decade or more behind radiology in the adoption of digital imaging into our everyday workflow? Well, there are several reasons that can be cited for that. First of all, native image format. The new radiographic imaging modalities, CT and MRI, that were developed were natively digital, whereas viewing pathology preparations or slides on a standard light microscope, still done every day today, is a strictly analog process. With respect to workflow, I mentioned that initially these digital images were reproduced on film so that radiologists could read them in a manner that they were used to reading radiographic images. But eliminating the reproduction of that analog image on radiographic film eliminates a step and therefore a significant amount of cost from the workflow. Whereas digitizing a pathology slide adds a step to the workflow and adds cost, both in terms of equipment required and personnel time. The distribution of images is different. Radiographic images are widely used by non-radiologic clinicians in their regular day-to-day clinical diagnostic efforts, whereas pathology images are rarely distributed for interpretation by clinicians. So there wasn't really a crying need to distribute pathology images outside of the realm of pathology itself. There are certainly differences in workplace requirements. CLIA has very specific requirements for where and how pathologists work. And I think if anything positive has come out of the COVID era, it has been the loosening of the requirements for validation of digital imaging so that pathologists could work remotely in a socially distanced manner. There are, by contrast, no real similar requirements for radiologists like CLIA that regulates the practice of pathology. Image file size is certainly also a consideration. Pathology images, usually color, are 10 to 100 times larger than radiology image files. That requires a lot bigger bandwidth for transmission of images, and it requires a lot of storage space. Thankfully, as I mentioned, storage keeps getting cheaper and cheaper, but it's still a real cost. And then finally, we have a significant difference between the fields of radiology and pathology with respect to image format. Radiology, early on in the process of adoption of digital technology, adopted a standard called the DICOM standard, and that standard is used by all software developers and equipment developers to develop radiology imaging equipment and formats. Whereas in pathology, we have no such existent proprietary image format, or standard, I should say, so that in fact, what has developed have been a lot of different proprietary image formats. There is, thankfully, an effort to develop some standards of image exchange in pathology, but again, this was an afterthought and not something that was adopted at the beginning of the process. Certainly, there are benefits of digital imaging for both pathology and radiology, things that we share in terms of this, in terms of common goals. One of which is that there's a lot less opportunity for loss of patient material and research material. So a lot less effort, a lot less risk of losing films and slides, and therefore your entire record if things are digitized and stored in that manner. Digital images facilitate easier distribution of images for clinical, educational, and research endeavors. We use digital images routinely in tumor boards and in CPCs. We teach students, residents, and fellows with digital images. In fact, I don't think our medical students even touch microscopes anymore during their medical school training. And then certainly in the research realm, the collaboration among investigators at different institutions is greatly facilitated by the sharing of digital images. Digital images also offer the opportunity for unlimited simultaneous access by multiple users. And this is useful in the clinical, educational, and research realms, of course. And then finally, and maybe most importantly for this group, is that digital images provide a format that can be used for additional computer-assisted analyses. One of the most important of these analytical methods that is being rapidly adapted is the use of artificial intelligence and machine learning in the application of analysis of pathology images. We can take a couple of different approaches here. We can train a computer to do what we do so that a computer can recognize the specific features of disease that we already know. We can teach a computer to count tangles and plaques and quantify the features of Alzheimer's disease in that manner. But maybe more important than this is the ability to use artificial intelligence and machine learning to discover new disease features, like the complex relationships that exist between different tissue elements, or the recognition of non-canonical lesions. For example, as was done by Marksberry et al. Early on in the recognition of dystrophic neurites as a critically important determinant of extent of cognitive impairment in Alzheimer's disease. So we have a lot of tools available to us now in the digital pathology realm. Many of these are going to be covered in subsequent talks in this series, and I'll try to highlight those. I understand that maybe some of these dates aren't etched in stone yet, so you may want to keep a lookout for future schedules. But selecting a slide scanner will be covered by Maggie Flanagan. There's a guide to slide scanners available now on the University of Washington NACC website. Aperio software will be delved into more in detail by Melissa Murray. We have a number of telepathology platforms available to us, which are not covered in this talk, but are widely used now in the clinical diagnostic realm in anatomic pathology. There are a whole host of analytical technologies available now. Halo software will be covered by Dirk Keene and colleagues in a later talk. Pete Nelson will talk about other methods of quantitative digital pathology. The QuPath software will be covered by Roddy Shahedzi and Tom Pierce. We have extensive availability of confocal, in vivo, and multiplex imaging, again, not covered in this talk. And then finally, artificial intelligence and machine learning will be given an overview by Brittany Duggar in a later talk in this series. So with that, I will wrap up my presentation. Thank you for your attention. I hope it was helpful, and I'll turn it back to Maggie to introduce our panelists.
Yeah, thank you so much. That was a wonderful talk. I don't think I had realized that Dr. Marksberry was such a pioneer in the digital pathology area. So that's neat to learn. So I just want to introduce our panels for today, and I'm really excited to introduce this group. I know most of them quite well, and we also have someone coming from a more cancer-focused neuropathology background, which I think will be great to get their expertise. So the additional panel members, in addition to Dr. White today, include my colleague, Dr. Brittany N. Duggar from UC Davis, who is Assistant Professor in Residence of Pathology and Laboratory Medicine, and the ADRC Neuropathology Core Leader. We've collaborated on a couple of digital pathology projects in the past couple of years, which has been really great. Dr. Peter T. Nelson from the University of Kentucky has been one of my mentors and colleagues for many years, and also a pioneer in the area of digital pathology. He's a professor of pathology and director of neuropathology, as well as the ADRC Neuropath Core Leader for the University of Kentucky. Dr. Sandra Camelo-Piragua is coming from a cancer background, as I mentioned. She's from the University of Michigan and is a clinical associate professor and neuropathology fellowship director. And then my former neuropathology fellowship director is going to be joining us, Dr. Dirk Keene from the University of Washington, who is an associate professor of laboratory medicine and pathology, as well as the Nancy and Buster Alfred Endowed Chair and Director of UW Neuropathology, and the associate director and precision neuropathology core leader at the University of Washington ADRC. And Jeanelle Azira Torres is the last panelist member for today. She's also from the University of Washington and is, I believe, our group's Halo expert, and she is a Keene laboratory research scientist. So yeah, take it away if we have any questions popping up or if you want to comment on the session. Okay, well, I can, I guess, kick us off here. One of the things that I thought was really interesting, and I'm kind of envious of the radiologists, is that they were able to standardize their format early on. And I know that personally, that's something that's quite a challenge for our group, in that when we scan slides in, you know, depending on the system used, it can add a very time-consuming additional step in order to convert the file to be able to perform our analysis. So I don't know if anybody has any commentary or insight into their experiences with that, or suggestions as to how that might be streamlined or optimized. Like, not everybody all at once.
That is, it's a big challenge, right? I think if you think about from like a research perspective, when you think about collaborators, maybe a good idea to set up ahead of time an idea of what kind of format you want to save your images on, to be able to be, you know, analyzed in a similar format. But sometimes it's not possible because it's something that you may do retrospectively. So the other thing is, like, you know, image conversion. It's a possibility, but depending on, again, what the focus of what the question is, it may be a challenge, right? If you have to convert and maybe decrease the quality of the image, or may not be able to use certain softwares to do, let's say, quantitative analysis or something like that. So I think, you know, trying to meet with collaborators and set up for a specific format ahead of time is key, but it's not always possible.
Yeah, and what do you specifically use for your format, if you wouldn't mind commenting?
Yeah, so it depends, right? So I think the problem is depending on the scanner, and we're just here talking about digital pathology of like scanning slides, right? So digital pathology is much more than that, but so some people use SVS formats, others convert to TIFF. We personally use SVS, but others convert to TIFF, just because some proprietary scanners that don't allow to export the images in their original format. So for example, the Philips scanner, you know, for the clinical point of view, has, because of patient information and all these kind of things, it may be difficult to de-identify or remove certain information that is going to be tagged to that image. So you may have to convert it and, you know, switch it to a different format. Other people even use DICOM, depending on the scanner. There are so many scanners out there that's I think one of the problems.
Thank you, Dr. Camelo-Piragua. I think you touched upon a very important point, and Dr. White, thank you for such a great presentation. One of the biggest things, and a lot of the people on this call are involved in the Alzheimer's Disease Research Center's Digital Pathology Working Group. If you're interested in that, feel free to email me. They can put the email in the chat. But we've been meeting now for over a year, and we're trying to talk about how do we standardize some of this stuff, or do we standardize some of this stuff? So even though you talked about some of these scanner formats, there's a whole bunch of other stuff behind the scenes. And so there's things like compression, metadata associated with it. What magnification do you scan in at? And so one of the helpful things that have been very good for us meaning I I'm very blessed to be a part of multiple collaborations within the state of California, as well as across the country. We sit down and we come up with those standardized protocols, just like how you would do with any experiment, right? Like, how are you going to decrease variables and this and that to get to that end? And I think that if people are interested in digital pathology, to have those deep discussions in the early stages of bringing on your slide scanner of, okay, hey, we're going to have immunohistochemistry slides. What type of magnification should we scan in? Should it be project specific? How should we do? And have these kind of at least somewhat guidelines, I think is helpful, because if not, it does turn into the Wild West. And then how do you aggregate that data if you want to do a bigger project? And I'll see if any of the other panelists want to chime in on that, because I think that's really important to understand, is there's a lot of different parameters, and have those deep discussions, not just with pathologists though. I think it's very important to bring on a diverse dialogue. So bring in your IT people, bring in computer engineers, because these are the people that are going to be helping you with data storage, as well as utilize those images as well. As much as I would love to say I code in Python, I don't. I have great collaborators that help me with that. And to have that open dialogue from that foundation really can help.
Yeah, I can. So a couple qualifications, right? So this is this whole field is really daunting to try to navigate. The way I see it, and we're getting into it in a more recently, unlike some of the panelists who've been doing this a long time. I mean, we're coming into it, and we're coming into it pretty big with respect to the different kinds of projects that we have. But the way I think about digital pathology is there's sort of two broad applications. One is day-to-day workflow in diagnostics, pretty much, right? And so you have GE and others who are really building platforms and applications so that there doesn't have to be a whole lot of consideration about, how you're going to set things up this day and that day. It's just it's a workflow, much the same that you would see in radiology. And then there's what Brittany's talking about, which most of us, certainly the panelists, are involved with, which is the research questions and having to really tailor what we're trying to do based on the scientific question. And in that setting, you want to have the options to control a lot of the different parameters. And I have to echo, there's no way that I can understand everything that's going on, right? That's why, like, Jeanelle is our expert who can answer all the crazy difficult questions. But at the beginning, middle, and end of your research project, you really need to have set up and understand and maintain the kinds of you need to really have it focused on what you're trying to get out of it. And if you don't do that, you're going to end up with an answer to a question you didn't ask, if you get an answer at all. So it's really complicated. I would just advise people selecting your scanner, which I think Maggie's going to talk about next time, selecting the software that you're using to analyze the images, and how you do that is going to have a big impact on what you do, but then also on your ability to share it with colleagues. And that can be really important. And so I think of these two bins, and then within these bins, there may be a third bin, which is education, which goes along the lines of sharing. I mean, we've had a lot of experience with junior neuropathologists or other trainees where we've been able to use the digital images. And with COVID, that's been a huge bonus, to being able to basically access this stuff remotely, as Chuck said. But depending on what you're trying to do, if you're just trying to go through day to day and have your computers counting things, then you set it up for one thing. If you have a big research question that's asking something very specific, like microglia associated with plaques and what their morphology is, then you need to really go in and think about what is the scanning, what is the magnification that you're going to need to do this, what is the software you're going to need to do this analysis. Do you need to apply a machine learning or AI of some sort to try to help tease apart what you're looking for? And so I don't even begin to have all the answers. All I can say is it is really complicated, and talking to people, and the whole point of this digital imaging work group is to try to help us as a community navigate our way forward until there become more and more standardized approaches, like in radiology.
That's great. Thanks, Dirk. I have a few questions, so I'll just kind of read them in the order in which they were received. So the first one is, how much of scanned material needs to be available at all times versus on demand? And how do you set up servers for this? Big time cloud storage? Question mark. So I don't know if maybe Brittany or
This
Pete?
is a great question, and Pete, please jump in too, because I know you have. Right now, I can just give you our own experience. So we have about a couple terabytes of data that we've been doing here at UC Davis, and this is a question of, you really need to do that deep dive of what are your needs. So to echo Dirk's aspect of, you have your day-to-day stuff, right? So you're signing out stuff, and then you have your research projects. So really, how much do you want access to that? And this is not just for digital pathology, but think of your own just research data. Like how much do you want that to be in, we call glacial storage. That's the long-term storage. These are just kind of, I think Amazon uses that term. So glacial storage. And again, pull in your IT people. Pull in, I mean, when we got our slide scanner, I was on the phone with our IT people from day one going like, hey, there's all this data. How can I store this in a feasible way? Can we partner together? And it does help to have that institutional support, not just from a standpoint of fiscal funds, right? But that staffing as well, because a lot of us think data manages itself. It doesn't. So you have to have that in it as well. And then to answer the question in the chat too, we do delve a little bit deeper in that slide scanner fact sheet, the PDF that is put in the chat. So we do talk about that and how to deal with that. But I think the biggest advice there is what are your needs? There's no blanket kind of thing of like, oh, well, this percentage or that percentage. Just think of your amount of usage and how much access. If you're doing machine learning processes, I recommend having your data as proximal as possible to whatever GPU clusters you have, because that can speed up your analysis. If you're trying to reach it through a server that's up in, say, like Quincy, Washington, or this and that, and then bring it down to a local, all that is going through the pipes, right? And sometimes you're trying to sell a watermelon down a McDonald's straw, even though your own building might have really strong Wi-Fi. It's that whole process. So trying to understand how you're going to be using it as well. And sorry for that analogy this early in the morning, but watermelon sounds good right now, maybe.
Awesome. Did you have anything else to add to that, Pete, or anyone else?
I made a little PowerPoint. I thought we were supposed to do PowerPoint. So like, if you want, I can sort of convey some of the stuff from the UK experience, but we can just do it by Q&A if you want. However, what would you like, Dr. Flanagan?
Yeah, if you want to go ahead and show a few slides, that would be great. And I think
Okay,
just
so this is sort of the UK experience. It includes some of the stuff that we've been talking about here, and I'll whisk through it. This a lot of the stuff that it's. Thank you, by the way, for inviting me, Dr. Flanagan, and it's a pleasure to follow Dr. White's wonderful talk and to be part of this nice panel. Thank you all for having me. The sort of traditional elements of neuropathology, then the digital pathology elements that have come in over the past decade or so. We got our machine back in 2011 from a large instrument grant. If anybody's interested in that, I'd be happy to talk to you about it. The pioneer here for us was Dr. Jana Neltner, who was at that time a neuropath fellow, and she developed algorithms with the ScanScope that enabled us. So what you see here is a tau stain on the upper left, and her algorithm could recognize both the tangles on the upper right, the neuritic plaques that are sort of orange on the lower left, and the total tau burden. On the top, you can see an A beta stain. You can see that it's labeled separately, the large, middle-sized, and small plaques. And so this is part of our workflow. Every single case that we have that comes through the ADRC is fairly systematically assessed in 8 different brain areas for amyloid plaque and tangle, neuritic plaque, and tau burden. This is our biobank coordinator. Shout out to Sonia Anderson. She is awesome. But every single one of our pathology reports for an autopsy has this chart for every single person, and those data are included as part of our normal workflow. So that's enabled us to make some research points as well. And so Dr. White mentioned that we have a history of quantitative neuropathology here that was spearheaded by Dr. Marksberry. And so Jana's work, Dr. Neltner, enabled us to compare the results of the manual counts versus the digital ones. And there were some sort of interesting differences I'm not going to go into here, but there's definitely serious advantages to the quantitative lack of floor and ceiling. We've also used the Alzheimer's disease neuropathological counts to correlate with genetic findings. I'm just giving you sort of an overview here rather than going deep into anything. But so that's for Alzheimer's disease, but other pathological phenotypes have also been assessed. This is TDP-43 proteinopathy. Here you see a hippocampus, and you can count the neurites, you can count the neural inclusions. And this was done mostly by a high school student at the time, Jean Borgal, who since moved on to college. And so basically I've sort of parasited onto a bunch of smarter people than me, and they've done a lot of cool stuff. This one was done a separate approach. So I've discussed real quickly looking at Alzheimer's disease phenotypes and TDP proteinopathy, but you can also look at other types of phenotypes. Here, there was a count of microglial phenotypes in a segmented hippocampus, and this was done by Adam Boxdotter, who's a PI here at the University of Kentucky, counting different subtypes of microglia that correspond to different disease types. And so finally then, in addition to things like pathological inclusion bodies or cell types, you can look at other things. And this is something that Dr. White referred to a little bit. You can look at other anatomic and histopathological features. And here we looked at, this is again, Dr. Neltner's work, looking at profiles of vascular profiles in the brain. And we optimized immunohistochemical staining. And it's something to keep in mind that if you're using archival material, that the layability of your antigen to fixation and other artifacts is very important because there's a definite garbage in, garbage out for the digital pathology. And we found that arteriolosclerosis was associated with the hippocampal sclerosis and the limbic predominant age-related TDP-43 encephalopathy phenotype. And so enabled us to generate hypotheses in addition to test hypotheses. And so I think it can be a very powerful set of methodologies in the neurodegenerative neuropathology world. And so I just wanted to sort of cruise through that stuff, and I will unshare my screen. But with regard to what the question that was at hand, about how you deal with technology and get AI and get all the storage, you have to, I think that there's a lot of center specific ways that you have to work, as Dr. Duggar was saying, how you have to sort of get the arrangements in place. But I think it's worth the investment, especially as we have well over, I think 100 terabytes of, because we have our whole brain bank is now essentially virtualized, and so that we can use that to fairly quickly do a lot of experiments in silico, and so that's nice. But in order to do that, we have to have a good arrangement with our IT folks and with people across campus.
I can add additional information regarding that question. It's important to ask you how big is going to be in the future your project, because that depends on if you choose on-prem services or if you decided to go for the cloud services. Right now, the cloud services are really amazing. They are very user-friendly. And the advantage with the cloud services is that the scalability. You don't have to worry about transferring your data or taking care of your data in some way, because those services are paid for the quote that you pay for those the cloud services. And also you have tons of backup options. You can transfer your images, you can move your images freely, and you can share your images with HIPAA compliances, and also to have like a certain independence from the IT departments, because sometimes in the universities, the IT departments are really busy attending other important stuff. So taking care of the cloud account, it depends if you want a huge material of investigation and research, and if you have tons of slides, so you don't have to worry to transfer the slides or buy more servers in the future. So you need to think really well, what do you want to do with your data before to enter into the digital pathology world.
Thank you. That's great. I have a few more questions here. So this is kind of a nice segue. So, this is some from Kate Schwait. Apologies if I'm pronouncing the name incorrectly. Hello, I'm joining Washington University ADRC very soon and very interested in AI as applied to image analysis of H&E and IHC stained sections. Who is interested in working on this? What kinds of grants are available for this? And how do you engage computer science colleagues? So I can comment on, I don't know of any specific grants for this personally, but others may. But I'm actually working on a grant that incorporates a lot of digital pathology just through the regular, NIH grant application process. And I think others have had success with that approach. But if anyone else on the panel has any specific suggestions of funding opportunities, I know you mentioned, Pete, that seemed more instrument focused, but if anyone knows of any image analysis focused grants, would be great to comment on. And also any tips on engaging computer science colleagues.
I can take the computer science colleague one because I've had a lot of great luck with this. I'm part of a large university, University of California, Davis, and we have a huge undergraduate body with many different majors. So I ended up getting in contact with the computer engineering department, and they have a lot of undergraduate students that need senior thesis projects. And so a lot of the things that, like the most recent one is a gray white matter segmentation project that I worked on with a student. Hey, think about how many hours we spend tracing gray and white matter to try to get all those detailed aspects for the deeper phenotyping. And that's kind of where I've done it. So I highly recommend, if anyone's part of a university, or even if you're part of a neighboring university, right? You might have some other colleges or universities nearby you, reach out. And this is a great way to get them also involved, because you have a lot of undergraduates that, might be going into computer science, but introduce them to healthcare imaging. There's such a huge need right now. And especially if you're digitizing stuff, how can they utilize this and that? So that's just from my standpoint, I've had really good luck with that. And then the student gets credit, and it's kind of a win-win situation, which is really nice. And then with respect to funding, I've been very blessed that we submitted an administrative supplement. And that's how we got our digital slide scanner. And in that, one of the aims was to do some quantification. So that's how we did it. And then Pete said about the S10. So those are just some mechanisms. Obviously, they're not getting, no grant is guaranteed, right? But just hopefully that helps a little bit. And then obviously institutional support. We did a survey about a year ago, and about 50% of people, I think that was the number, 50% of people who have some of this digital slide scanner had institutional support. So it's that thing of what can you pull together? So yes, you have pathology, but there's also some other stuff like dermatology, this and that. Like, how could you pull together multiple resources at your institution to have that common goal? And that can always help as well.
Great, thanks. I have a couple of questions about how slides are shared. Are there currently places where they can be shared? Specifically, there's a question asking how the ADRC program will make available digital images to the research community for projects. So if anybody wants to comment on that, that is a work in progress. I can start by saying that. And I know that the Halolink specifically is one option that we're looking into, and maybe Dirk and Jeanelle have more input and can comment on that. I haven't personally tried it yet.
Sure, I can start. So Halolink is just one of many different ways that you can basically open up your digital archives to people, to share with people. There's a lot of different mechanisms to do that. I think to emphasize basically what Maggie alluded to right now is this is a work in progress. And I think that is one of the driving forces behind this working group that Brittany and Melissa are leading to try to figure out, right, how are we going to try to make this this really important information available. I mean, you know, in theory, this is going to really open up the opportunity for us to all collaborate, to share the data and tissue resources that we have with people who may not otherwise, you know, have the bandwidth or the resources or whatever to actually request a bunch of slides to send out or things to stain. And so I don't think there's a right answer right now. It's kind of the old Wild West with respect to how this is done. You know, I guess I would ask Pete, because Pete has a big digital archive, right? So when people want to share those images, how do they do it? And the way we're going to do it is with this Halolink, where you can give people access, and then you can come in and access things. Or in our work with the Allen Institute, right, where they have specific needs, we may actually send them or transfer the actual files. But as you've heard, that is not trivial, right? This is a lot of information. So I would ask the panelists who have more experience with this than us, what is the strategy when people want to access your digital images for research?
Well, we don't really have a strategy for doing it to people outside our university. So that's stupid and bad and backward and everything else. But it's just we don't, and that is something that is a developmental aspiration at this point.
The other situation that you need to take into account is that the people that you are going to share the images with, they need to have access to the software to read those images. So if you end up acquiring Halo, you will have like a certain amount of users that you can allow to enter and see your images and part of the algorithms and data that was produced by the software. I know that Aperio system also has that ability with image scope, but so it depends of which software do you use that you can allow other users to get access to your images. The situation is that you need to know the rules that applies to your on-prem services at the university, because they need to follow the policies of HIPAA compliances and also the access through the university that needs to be authorized.
Yeah, so I think another thing in terms of collaboration is like, data transfer agreements between universities. So again, everything comes down to the question, the research question, who's going to be part of the team and how you're going to do it. And things can change, obviously, but it's something that one has to think ahead of time. And those data transfer agreements are very important to set in place from day one, because it can take time and each university may have different thresholds of what can be shared, how can it be shared, where it will be stored. And I'm not sure how it is for ADRC, because, to me, ADRC falls into a middle ground between clinical and research, because we are still serving a purpose for the patient and the families, but we are also taking the information to develop and move knowledge forward, right? So it's different. Like, how are you going to scan your slides? You're going to scan it as part of clinical and use your clinical scanners? And if so, will they have identifiable information? Or are you going to use your core facility or a research scanner and give them the identified labels for your research purposes? Those are things that all have to be taken into consideration. And finally, I will say something, clouds, like certain universities may have cloud storage that will allow people to get in and share access, or as it was mentioned before, Amazon or Google, they all have these things. But again, back to personal information, make sure that it's not there in the public. And then kind of plan for one project or your project, with 30 or 40% extra space. And eventually these will have to go to a cold storage, as it was mentioned before, because it's impossible to have everything on the cloud at this point, really. So eventually you will have to take down your old projects and move it to a cold storage. And if somebody needs it, you pull it out from your cold storage and then share it. But it's at least for now.
Thank you. Yeah,
really quickly, we are, because I'm seeing some of the comments, for us, we are sharing digital slides and we're treating it just like a tissue request. So people have to fill out a form. We do the DUA, and thank you for mentioning that, Dr. Camelo-Piragua. That's very important, is the DUA. And the biggest thing, we kind of put it on the investigators now that are requesting for how to transfer images. There are HIPAA compliant transfer services, so we actually send them the whole file, the digital file. And there are some ways for those people who don't know, and feel free again to email one of us. There's some great software, if Dr. Pierce is on, that can black out a lot of the HIPAA data on that metadata on the slides. So there are some ways to share if slides are already scanned in and identified. So we share all de-identified data to that. That's part of the DUA. Hopefully that's helpful for people. So yes, we are sharing slides here at UC Davis.
Brittany, if I could just add though, I think some of the questions are really driving at what does the future look like for the ADRCs, and I think future webinars will probably, I mean, this is the point of this working group, is to try to figure out should we and how do we go from each of the individual centers managing their sharing like we do with tissue to the possibility of some sort of a more coordinated effort across the centers. And I think there's no right answer to that at this point, and that's why we're all working.
Well, and is it a mandate? Like, think about the latest, not to get too distracted, but like our biorepository core that was just added. So let's go back to what we actually need to do as cores and what we would like to do. I think that's a really important thing as well, because as you know, with resources, we have to make our resources stretch farther and farther every time. Like now it's adding our tag stuff, adding this, adding that. So it's always all this adding, but where's the added resources to help with that? So I feel like that's a good point to maybe segue into the last. And happy International Women's Day, everybody. I'm going to give that shout out too.
All right, so we should probably wrap up because we're at the end of the hour. I didn't get a chance to address a question that was asking about studying the validity and reliability of images produced by different proprietary machines and imaging processing processes, but I can address this at the next session, which is focused entirely on side scanners. So that will be great. So I just wanted to thank you, Dr. White, and all of the panelists today for participating. I think this is really exciting and a great kickoff session. I look forward to all of the other more comprehensive sessions on each software option, etc. And I just wanted to announce our next session will be on May 10th from 11 to 12 Eastern Time. And I will be presenting on selecting the best slide scanner for your group. So I'll do an overview, introduction, and then talk about some of the specific factors to take into consideration when you're selecting your scanner. And then, yeah, we'll have, as I mentioned, series second Monday of each month, most months, and we will distribute the finalized agenda for the entire year in the upcoming weeks. So thanks again, everybody.